Okay, we're looking at a pre-lab here today on Mendelian genetics. This is a handout, so you need to have a copy of this handout before we begin. You may want to pause this and go print that off. I don't know how many pages there are, but quite a number. We're going to try to do about half of this, and then that way in class we only have the other half to do. That's the only way I see to get all of this done in the time frame that we will have to do it. So again, it's topic 10, Mendelian genetics. They do tell us that it is imperative that you understand meiosis before attempting to do genetics problems, because these are certainly, that is something that relates directly to genetics. So on the discussion, they talk about the individual who developed the basic process of genetics, and these are the basic principles that we use today. This guy was an Austrian monk. His name was Gregor Mendel. He was a gardener for the monastery, and his plant that he studied was the pea plant, the common pea plant. And he was trained in mathematics, so he kept lots and lots of statistics, and with that then founded what we consider to be modern day genetics. So at the time it was pretty amazing, considering they did not even know that DNA existed. Okay, so and that was in the 1800s, little kind of middle to late 1800s is when he did that. Okay, what you'll also find, you'll see several definitions which are important. On the last lab exam, you will have several definitions, and then of course you'll have to tell me, for example, you'll notice the first one is qualitative and quantitative cell division. Meiosis is a process by which all cells of the body are produced except the gametes. So that's called a mitosis. And so I will give the definition, and then of course you'll have to tell me that that is mitosis, and so forth. So we have several here. Cytokinesis, that just means division of the cytoplasm. Kinesis is division, and cyto means cytoplasm, so division of cytoplasm. That's when the cell pinches into two units is what you have. A meiosis, a reductional division, of course that's where we, the process by which we use in order to produce the gametes, the sperm, the egg. Spores, if you're happen to be a fungus, it would be pollen grain if you're fairly an advanced plant, if you're an advanced plant. So there are different types of gametes out there, but anyway, for animals, we typically use sperm and egg. And then the chromosome, of course, is really, that contains the DNA, but the DNA is really quite long, and in order to package it, the DNA wraps around proteins called histones. So as long as you have the DNA associated with the proteins, then together we can refer to that unit as a chromosome. So it's just DNA that's wrapping around protein histones is really what that is. Now, in mitosis, as well as in meiosis, we have talked about that cells replicate, and when they replicate, they remain attached to each other. And so let's just pretend that we have this one chromosome, and of course it's going to replicate, and so as it replicates, we do have two copies. Well, each copy of that is referred to as a sister chromatid, and they're exact copies of DNA. They carry the same genetic information, and they're the same length. So they're exactly the same. Now, at some point, whether you're in mitosis or meiosis, you do pull the two sister chromatids apart and pull those to different poles. But those would be the sister chromatids, which will be when our DNA replicates. Okay, the chromatin, if you were to see a cell and you were to see it in an interphase, and an interphase, of course, the chromosomes have not been condensed in. At this point, they're really not visible. So you can't really make out individual chromosomes, and so you just see a mass of material within the nucleus, and that would be all the chromosomes that you have within that nucleus. And like I say, they're not condensed. You can't make out any individual chromosome. So at this state, it's kind of a loose, uncoiled state. That's what we call the chromatin material. that is an interphase. Now, once we move into a prophase of mitosis or a prophase 1 of meiosis, the chromosomes will actually condense and become visible, but in interphase they are not, and they're referred to as chromatin. And that would be all of the chromosomes together. Now, we've also talked about you receive 2 copies of each chromosome. One of those, of course, you get from your mother. Well, let's say here that the blue one would be the father's chromosome. And same thing with the chromosome pair number 2. You get 1 chromosome pair number 2 from the mother, and you get 1 chromosome pair number 2, or 1 chromosome number 2 from the father. So again, the blue one would be the father's chromosome, and the red one would be the mother's chromosome. And you also get 2 3s and 2 4s and 2 5s and 2 6s. But for we refer to these as homologous chromosomes. So they're and they carry the same traits. For example, if we had a trait for eye color, let's say blue eye color on 1 chromosome, the other chromosome might carry the code for, well, the capital B would be for brown and the small b would be for blue. So that would be the actual gene for eye color, and of course the trait in that example would be eye color. So homologous chromosomes, they actually carry the same traits on those chromosomes. So we have a total of 23 pair of chromosomes. Now, going back to homologous chromosomes, let's go to the number 1 from the father and the number 1 from the mother. What you really end up getting here are several genes, any one gene, but if we're wanting to make this simplistic at this point, we'll just talk about a single gene. So let's say like gene for eye color is pretty easy. So we could have had the gene for brown eye color donated from the mother and the gene for blue eye color donated from the father. So we have 2 different forms of the gene. You have a small b and a capital B. But when you write that, you typically always write the dominant gene first and the recessive gene second. So that's typically how you write the gene actually being eye color. The 2 different forms, in this case, one is for brown and one is for blue, those are called alleles. Gregor Mendel actually called them factors, is what he called them, but of course later they changed that and gave that a more scientific name, and that's the name they chose, the alleles. So those are different forms of a gene. Although you do hear the term allele sometimes referring to, say, like if one parent donates a code for brown eye color and the other parent donates a code for brown eye color, they call these alleles. Well, technically that isn't correct, but they do use that terminology. They're not alleles because they're not different, but again, still, it is used, it's commonly used, so you'll see it used time and time again. Same thing with this. You could have one parent donate a code for blue eye color and the second parent also donate a code for blue eye color. Well, technically they're not different, they're both for the same color, but so technically they really shouldn't be called alleles, but you do hear that term used. So we have 2 blue-eyed alleles is the way that they would actually describe that. Now, you know, they take a little liberty with some of the terminology that they do use. They'll give you a definition for it, and then they'll turn right around and break the definition, which I always find pretty interesting. Of course, then what we're looking at again on those chromosomes there, you see the blue one and the red one. Well, the gene, the genetic code for either blue eye color or brown eye color, we have at a particular location on those chromosomes. And further down on the gene, you may have a gene for some other trait. Say like some people, when they clasp their hands together, you may have the right thumb on top, and on some people, the left thumb would be on top. So, you have a gene for that particular trait, and it's at a very specific location. So, the location to find a particular trait, say like, for example, eye color, that location is what we call a locus. Locus just means location. Same thing here. So that's the location for eye color that we have. Here you have a location for whether your thumb would be on top, right thumb would be on top when you clasp your hands together, or your left thumb. So the area on the chromosome where that gene is located is just referred to as a locus. So that's just another term for really location. Okay, let's go back to alleles. So if we had the true sense of the word allele, would actually be, say, a capital B from one parent and a small b from the other parent. Well, that would be, those would actually be alleles. But technically they refer to those as heterozygous alleles. Hetero meaning different, so alleles are different. One is for a dominant situation and one is for a recessive situation, brown being dominant, blue being recessive. Now, if both of those genes are identical, they call it homozygous. And again, these are homozygous alleles, which, you know, that actually is really quite contradictory because, you know, saying alleles, that means they're different, but then using the term homo tells us they're the same. So both of the genes are the same. They're both for blue eyes, but they use the term homozygous alleles. In this particular case, this would be referred to as homozygous. So they're both the same, and they're both recessive genes. So this would be homozygous recessive situation, both being recessive. And then, of course, in this situation where you have a brown gene donated from each parent, one from the father, one from the mother, then again, this is homozygous, and they're homozygous alleles, but they are then technically going to be homozygous dominant, both being the same, but both being dominant genes. It's dominant. So again, there is quite a bit of terminology that does go with genetics, and that's probably the most difficult part of genetics would be the terms, those lined up in your head. Now, if I had two individuals that have brown eyes, so this person obviously is going to have brown eyes, the mother and the father both donated the code, and let's say that individual mates with another individual who has brown eyes, well, you know, the only possibility out of that would be, well, let's produce gametes here first. So the only gamete possible that you can get from a person with brown eyes, the gamete, of course, is going to be half. They're not going to have both of those chromosomes. Say like this is with chromosome number 1, and it has a code for brown eyes. And then the mother's chromosome number 1, which also has a code for brown eyes. All right. So if we're going to form a gamete from this individual, well, the gamete, this is a diploid, 2 copies of each number 1, 2 number 1s, and 2 number 2s and 2 number 3s and so forth. So that is actually a diploid state. But if you're going to form a gamete, you're only going to get 1 chromosomes. You could either get the blue one or you could get the red one. But let's say if we're producing, say, for example, a sperm, then we'll just get 1 of those. In this case, we'll pretend it was mother's. It could be the mother's number 1 chromosome, but we'll say it's the father's. Same thing for this other individual. This is a diploid state. It's 2N. So we could get either 1 of those. Again, we'll pretend that they're on the number 1 chromosome. I don't really know which chromosome pair contains the genetic code for eye color. I've never bothered to look that up, but you have a computer probably handy, so I think you could look that up pretty easily. So the egg, let's say, is going to get the code from the mother, which would be for brown eye color. So if we had a union of these two, then of course the offspring are going to get 1 code from the mother and 1 code from the father. So once again, we reestablish the diploid state. So we get a capital B from one parent, that being the father, and a capital B from the other parent, that being the mother. So we're back to a diploid state, 2N, and it's still homozygous dominant. The sperm, of course, only got one of the two chromosomes, so the sperm is going to be haploid. The egg also is haploid. So by having a fertilization, we've reestablished the diploid state. The other thing to take away from this is that if you have an individual that is homozygous for a trait, like capital B, and the other individual is homozygous for a trait, capital B, the only possibility you could possibly get out of that mating would be capital B. And that being the case, these are what you call true breeding. So Mendel happened to use true breeding pea plants in his experiments. So that was very smart. He bred, say, like a true breeding plant that was very tall with a true breeding plant that was short. Okay, so true breeding for tall and true breeding for short, meaning that if you crossed a tall plant with a tall plant, you would get all tall plants, and if you crossed a short plant with a short plant, you would get all short plants. But if he did a cross between those two plants, that's and there's no such thing as heterozygous dominant or heterozygous recessive. It is just heterozygous. But again, if you cross a tall pea plant with a tall pea plant, you're going to get a tall pea plant, and if you cross a short pea plant with a short pea plant, you're just going to get short pea plants. But if you do a cross between the tall and the short, all of the offspring are going to get one of the capital T's and one of the small t's in the gametes. So all of the offspring would be capital T, small t, in this case, heterozygous. So how he lucked into thinking that he needed to choose the tall pea plant in his cross and the short pea plant, which would be heterozygous, that would be homozygous for both cases, which means that if they crossed the tall pea plant with the tall pea plant, it would be resulting in offspring that had that were tall pea plants. How he knew to choose true breeding plants, but he did, and that turned out to work to his favor. So he ended up with lots of statistics that he could use, and here again, it turned out to found modern day genetics as we know it. So it ended up being a wonderful thing. Okay, now let's skip on down to what we call genotype and phenotype. Now, genotype, you're looking at it. Say, for example, an individual that gets two capital B's, one from each parent, well, that is the genotype. It's what genes are present. Or if an individual gets two small t's, that is the genotype of that individual. Or two capital T's, that is the genotype of the individual. However, phenotype is what you would physically see if you were looking at that individual organism. Well, in this case, the phenotype would be tall pea plant. So that is phenotype. This plant would be short. That is the phenotype. That would be the actual physical expression, what you see from the genotype over here. This individual, what you would get out of it. And this individual also, this is your genotype right here, would also have a phenotype, which would be brown eye color. So genotype would be what genes are present. Phenotype would be what the physical expression of those genes would actually be. So that's how you would read that information. Okay. We also have, we understand, a total of 46 chromosomes. So we get 2 number 1s and 2 number 2s and 2 number 3s and 2 number 4s and so forth. So this goes all the way down to 2 number 22s, which would be very, very small chromosomes. That's how they label them. So the number 1 pair of chromosomes are very tall, the number 2 are shorter, the number 3 are shorter. the number 4 even shorter, and by the time you get to pair number 22, they're very short chromosomes. So all of these, which is 22 pair, so that's only 44 out of our 46 chromosomes, these are called what we call the autosomes. And autosomes are chromosomes that carry genetic information for traits other than sexual traits. So whether you would produce a penis or a vagina, that information is not carried on an autosome. That information would be like hair color, eye color, whether you're 6'4", 5'2", you know, how much hair do you have on your knuckles, and the list just goes on and on. The size of your spleen, you know, so forth. So those carry traits for anything other than sexual information. Now, we do get one pair of sex chromosomes, which is the 23rd pair. Now, if you get one long chromosome, we call that the X chromosome, and a really short chromosome, which we call the Y chromosome, then this individual would be a male individual. So 2 1s, 2 2s, 2 3s, all the way to 2 22s, those are the autosomes. But the X and the Y, those would be the sex chromosomes, and they determine the sex of the individual. Obviously, they're going to determine the sexual organs that are produced, and I think most of us would know the difference between a male and a female. That's not a difficult situation. Now, if, of course, a person is female, they really get 2 X chromosomes. So they get 2 long X chromosomes. Well, so this kind of sets up a really interesting situation where, let's say, there are some traits that you find on the sex chromosomes that have nothing to do with really the sex of the individual. For example, color blindness. So if I have a male that has, we'll say, a small c here, this male, because you don't have that information on the Y chromosome is much shorter, so there's no complement to it. You don't have a complement. So this is a male that would be colorblind. Now on the female, let's say that we get a code for color blindness, but because she has a complement to that, then she could have a code for color vision. So this individual would be a female with the 2X chromosome, but this would be a female who has one genetic code for color vision, one genetic code for color blindness. So the dominant gene will mask the expression of the recessive, meaning that you will only be able to observe the dominant gene. So this individual would actually have color vision. They would say this individual is a carrier for color blindness. So that's another way to put that, because this is a female individual who could, if you'll remember, to produce the egg is either going to get this X chromosome here, or it's going to get this one. It's not going to get both of them, because if you remember, the egg is just haploid. It's only going to get one of those X chromosomes. It's going to get one number 1, one number 2, one number 3, one number 4, one number 5, and it also gets one number X, one of the sex chromosomes. But it could be either one, the one for color blindness or the one for color vision. I happen to pick up the one for color blindness from my mom, who's a carrier. So in her egg, I got the color blindness code. So that was the X chromosome that I received, which is right here. The Y chromosome, you always get that from your father. So I got that from my dad. So this one I got from mom. The X, the Y, you always get from your father if you're male. And so obviously I have color blindness. And, you know, it doesn't really have too much effect on my life, other than I'll have no idea what colors I'm wearing. I have some color vision, but some of it's not accurate, which is interesting. Anyway, it's certainly different than most people. Most people don't have color blindness. It's very rare for females to have color blindness, and let's talk about that for just a second, because the only way that a female actually can be colorblind, you get 2X chromosomes. Now here again, remember, you do get 2 1s, 2 2s, 2 3s, 2 4s. You get the entire 44 autosomes, but you get one pair of sex chromosomes. Well, if her father donates a code for color blindness and her mother donates a code for color blindness. So the sperm has an X and a Y possibility. So if this parent, if the father donated this chromosome right here, well, the father had an X, which let's say had the color blindness code on it, and had a Y. Well, if he happened to donate the X to the sperm, then he was donating the code for color blindness. And the mother of this individual could have been, because she has two X chromosomes, she could have had the gene for color blindness and then also the gene for color vision. Okay, so she's heterozygous for that, and she's a carrier. Well, instead of the egg's going to get one of those, it could have been either this one or it could have been this one. Well, let's say it was this one right here that she donated. Well, this female actually picked up both of those, one from each parent. The X from the father and the X from the mother, and both of those were carrying the color blindness gene. So, in order for a female to be colorblind, she actually has to pick that up from both parents. So that's pretty rare. I'm not saying it doesn't happen, but it's certainly not as common as a male having color blindness, because he only has to pick that up from his mother. The Y chromosome will never have it. Like I said, it's just too short. It cannot have that. Okay, so X-linked traits. You're looking at those right here. Actually, this is an X-linked trait. It's a trait that is found on the sex chromosome, and in this case, specifically on the X chromosome, and it has nothing to do with the sex of the individual. This just happens to be a gene for color blindness. So it is an X-linked trait. It has nothing to do with the sex of the individual. Okay, it just happens to be on the sex chromosome. All right, now let's talk a little bit about synapses. If you remember, because we have discussed this, that during a prophase 1 of meiosis, so we're doing a reductional division of meiosis, you're going to have a number 1 from the father, and you're going to have a number 1 from the mother, and a number 2 from the mother, number 2 from the father, and so forth. But during prophase 1, these are actually going to join up. They will join. And let's go ahead and replicate those. So in interphase, which occurs before this prophase 1, in interphase, the number 1 chromosome from the father replicates, and the number 1 chromosome from the mother replicates. So when they join, that is called a synapsis, which I find real interesting, because the mother and the father's chromosomes actually pair up. So the ones pair together, the mother and the father's chromosomes pair. So every time you actually produce a gamete, your mother's number 1 chromosome is pairing with your father's number 1 chromosome. So it's sort of interesting that the chromosomes donated from each parent are still pairing. So in order to produce you had to have a pairing. And then even when you produce gamete, your parents' chromosomes are still doing the pairing. That's exactly what's happening there. So it's kind of a very interesting concept, the way that actually works. And the other beauty of this process is, if you'll notice, what I've done there is I have made a crossover, meaning that, well, let's say the two blue exact copies of chromosomes are called actually chromatids, sister chromatids. The two red ones are sisters, but if I cross a red and a blue right here, that point where they cross, well, that's called a crossover. And what happens with crossovers is they can exchange genetic information, and typically they do. So what you end up getting at the end of that crossover is you get a piece of, say, your mom's chromosome on your dad's chromatid, or let's say a piece of your mom's chromatid on your mom's chromosome, and a piece of your dad's chromatid on your mom's chromatid. Okay, so we've created a genetic variant out of that. So they've exchanged little pieces of genetic information. had they not joined together in a synapsis that we had happening in prophase 1, then we would not have had the exchange of genetic material that you see here, and we've created a genetic variant which we hadn't had before. And crossovers can occur say like between this point and this point, but they could also occur between this point and this point. So you can have several crossovers even on any one pair of chromosomes. So it does create lots and lots of variability, considering we're talking about a total of 23 pair of chromosomes that have the ability to do some crossing over. So you get a tremendous number of possibilities in terms of what the offspring will really be. So if you produce one child, you can get one genetic combination, but if you produce a second child, you know, it certainly will not ever be an exact copy if it were, of course, produced at a separate time. Now, sometimes twins do occur, and when twins occur, really, that's you have one zygote when the sperm meets the egg, and that zygote will begin to divide, and when it divides, you end up getting, say, like, initially, of course, you get two cells. You take one cell and you divide it into two. If for some reason those cells do not stay together as a unit and they separate, well, then each one of those cells will begin to divide, and you end up getting exact masses, which eventually become infants, you know, after birth, and they're exact copies of one another, and that would be what you refer to as identical twins, and that's how that happens. So that of course does happen. It's a wee bit interesting, but to me it's not all that interesting. I think it's interesting that you might have, say, another individual on this planet who is an exact clone of you genetically, an exact clone. Of course, you know, their experiences in the world may be different than yours, so you know they would be, you know, socially and mentally, emotionally, they would be different. But as far as the genetics of the individual, that would be the same. But then, you know, we always have learning that occurs during the course of a lifetime, and that is going to always be different between two individuals. So that certainly changes, even though they may be genetically identical. They would not be identical in all regards at all. Okay, so all of the things that we've really looked at to this point have been complete dominance. Going back to that, say for example, you get a genetic code from one parent for brown eyes and a genetic code from another parent for blue eyes. Well, the brown is completely dominant. It completely masks the expression, covers the expression of the blue eye color, so brown is dominant. And so everything that Gregor Mendel worked with was actually complete dominance. He didn't work with anything else. Now, there are some other things that we will see, like incomplete dominance and codominance, and when we get there, we'll actually talk about that, but we'll wait till we get there in this handout in order to do that. Okay, so again, lots of terminology, and it's just part of it. I know it's not certainly a simple thing, and not certainly simple in the time frame that we have to work with at all. But it's a challenge, and you know, I'm always up for a challenge. You know, throw me one. We'll handle it, and we'll do the best we can with it. We may not do absolutely perfectly with it, but we'll give it our best shot, and that's all we can do. We can't do better than that. So just give it your best shot. Move on. Life will continue, and you'll be a better person for having made the attempt. That's the way I look at it. Okay. So you see a Roman numeral 1. It says monohybrid problems with complete dominance. When they say monohybrid, that just means a single trait. You know, like eye color, that's a single trait, or hair color, you know, or how tall a person is, or you know, the hand clasping, whether the right thumb's on top or the left thumb's on top. Those are individual traits. So that's what we're working with, is monohybrid or single trait problems. Biology books at one point started talking about those as single trait. That's the way they listed them. And that was something new. They had always talked about monohybrid, and then they started going single trait problems, which I liked much better. And then they went back to just calling them monohybrid problems. So they do that from time to time. Switch terms and then go back to the old term, and then later on switch to the term that they had switched to before, and it's kind of a little game they play. I think mostly it's just so that they can sell books. It's basically why they do it. But we just play along with them. I don't guess we have much of a choice. But I really would rather call those single trait problems. We're only working with one trait. So they talk about garden peas. They have the interesting thing about a garden pea, it has both the male and the female sex organs on the same plant. So it self-fertilizes. It can self-fertilize. There are animals that can do this. Actually, a tapeworm can self-fertilize. It has female parts, and the male can fertilize the female. And that's not very common in the animal kingdom. It is certainly much more common in the plant kingdom, but it does happen in the animal kingdom. But a garden pea is one that self-fertilizes, and it almost always does that. So really for Gregor Mendel to cross, say, a purple flower pea plant with a white flower pea plant, he had to physically do that. It wouldn't normally happen in nature. So he had to take the pollen, which is the male gamete, from a purple flower pea plant and actually pollinate or fertilize the female part of a white flowered pea plant. And so he physically had to go into the flower to make that movement of that pollen. And so, he began to mess around with things, which normally that wouldn't have happened, which I find interesting on a different level, which is, okay, here you have a monk, and he's doing things with living organisms that weren't designed to be done. Well, God's the one that chooses what happens with living organisms. So, he didn't design them to do that. The pea plants, they weren't supposed to cross-pollinate. So here you have a monk who's actually playing God by crossing those two plants. So I find that, and I do think that God directs most everything. So here you have a monk who's a man of God, who's chosen to show us the basis of genetics. Well, when it comes right down to it, genetics is how we prove that evolution occurs. So I think that was planned. That was planned from the very beginning. That a man of God would show us how evolution occurs, and that evolution is certainly something that he designed and he wants people to understand. Now, of course, that's my take on it. You may have a different take on it. That's cool. I don't have any problem with people having different opinions. But I tend to look at things probably a little differently than probably most people might look at things. But I find that interesting. Okay, so let's see. This is exactly what he's going to do. Is do a cross, again, purple with white, and he ended up getting hybrids out of that, basically heterozygotes. He'll have a dominant for purple and a recessive for white. It's the plants that he created. Okay, on the next page it says, suppose one gene has alleles capital A and small a. Well, we don't know what the A's are. They're not defining that. A lot of people like to say, well, what is the A? And I go, well, okay, you got big apples and little apples. Okay, does that help you? The plant's either going to make big apples or it's going to make little apples. If you would like to use that, that works fine. I don't need to actually define the A. Now, we can call it anything we want to, but big apples, little apples, that works. Okay. So, of course, most organisms are diploid, so they have homologous chromosomes. And the alleles may be different on each chromosome, like one for big apple from the father and the small apple from the mother. Obviously, that plant would produce big apples since you got one for big apple. Again, from the father, and then you get, so we'll put a capital A on the chromosome from the father, and then we'll put a small a on the chromosome from the mother. Well, since the big apple we're saying is dominant, then this plant or this apple tree would grow up, produce big apples. All right, so that's just kind of what we're working with here, big apples, little apples. Okay, so, and since we're diploid, if you'll notice, they give us a diploid genotype. Like, for example, if the father donated the code for a big apple, so the capital A, and the mother donated the code for a big apple, let's say another capital A, then, you know, if you take this through meiosis, and kind of like to just do that physically. So let's say that we have a big A on the chromosome from the father, and let's say we have a big A from the chromosome from the mother. Okay, so remember, they're going to replicate. So the red one would replicate, so it's also going to replicate the big A as well. And then also the blue one would replicate, so we end up with two sister chromatids of the blue and two sister chromatids of the red, and both, of course, are carrying the big A code. Well, during meiosis, during the first meiotic cell division, those two chromosomes are going to separate. The homologous chromosomes separate at anaphase 1. And if you're kind of unsure of what we're talking about, go back and look at meiosis. So it's homologous chromosomes that move to the poles at anaphase 1. So really we're going to get a cell that ends up with one chromosome, although it does have two sister chromatids, both of those with the capital A, and that one was from the father. Then you get another cell that does have two sister chromatids making up that one chromosome, and both sister chromatids carry the capital A. And then you divide the cell. So we end up with these two cells. Okay, now during the second meiotic division, that's when the sister chromatids will separate, or sometimes they say segregate, either way. So if I pull those two sister chromatids apart and divide the cell, I end up getting two cells. This one will have a capital A on it, and this one will also have a capital A on it. And then if I divide the second cell, because again we do get two cell divisions, a meiosis 1 and a meiosis 2, then the sister chromatids will separate, so this cell will get a capital A in it, and this cell will get a capital A in it. So if you'll notice on your handout there, the diploid genotype is capital A, so that's what this is, capital A, capital A. And then you get four possibilities. You have four blanks. And in all four blanks, this is a capital A, this is a capital A, this is a capital A, this is a capital A. Now, of course, these two really were donated from the father. These two were donated from the mother, which will be the red ones. But it doesn't really matter. The gene is for Big Apple, whether it came from the father or the mother. It's really not significant. Okay, so, but if we had started with, let's say, for example, with two small a's, so the father donates a code for a small a that will, of course, replicate. And then the mother also donates a code for a small a, small apple. So it'd be a tree that's going to produce a small apple. All right, so the first thing you do is meiosis. We're going to separate the homologous chromosomes at anaphase 1 and divide that cell. So the chromosome from the father will end up in one cell, and the chromosome from the mother will end up in then the second cell. We've separated homologous chromosomes, and then we go through meiosis 2. So it divides a second time. We do have two cell divisions. So one cell divides into two, and then these two cells divide a second time, called meiosis 2. And on the second cell division, the two sister chromatids are going to separate. So what we end up getting is that is now was a sister chromatid, but once they separate, they're actually called chromosomes. They're called daughter chromosomes, technically. So these were sister chromatids, and now they pulled apart, and they're daughter chromosomes. So if you start with a cell that had a small a and a small a, at the end of that you actually produce 4 cells, and then all of those would carry a small a in all 4 cells that are produced at the end of meiosis, 4 gametes. So those might end up as sperm, and one of those might end up as an egg. Okay, so then you actually have another one where we have a diploid genotype where you get a capital A on one chromosome, and we'll go ahead and replicate it while we're here. and the mother's chromosome, we'll say, has the small a, and we'll go ahead and replicate that. So small a, small a. Then, of course, what we do, we go through meiosis 1, divide that cell, and you get 2 cells out of that. This one, of course, carries 2 capital A's, and then the second one is going to carry the 2 small a's. So we've separated our homologous chromosomes. That happens at anaphase 1. And then we go through a second cell division, which is meiosis 2. So one more time. We divide the 1 cell into 2 cells, and then we divide the 2 cells into 4 cells. So we end up getting 4 cells at the end of meiosis. And then each one of these is referred to now as an individual chromosome. They were sister chromatids, but we pull them apart, and then they're daughter chromosomes. And we do the same thing with the 2 sister chromatids that the mother donated. And so all 4 cells are complete, and the top 2 have the capital A, and the bottom 2 have the small a. And those would be the gametes. So our original genotype was capital A, small a, and when we're finished going through the cell division, we end up with 4 gamete possibilities. 2 of those would be capital A, and 2 of those would be small a. So again, all I'm trying to do is relate meiosis directly to genetics, because there is a direct link, and so it's really good to connect those 2, and that's why we do that. Okay, now if you look at number 2 on the handout there, what you're seeing is it says during fertilization 2 gametes fuse. Well, we understand sperm and egg, or pollen and egg, or whatever it is, they're going to fuse together. So if I have a small a and a small a, those are gametes, say 1 sperm, one is egg, and they're going to fuse together, plus then of course the other small a, and those are the haploid. Obviously those are haploid gametes. They only have 1 of each chromosome. In this case, we're just looking at 1 chromosome. And so we restore the diploid state, which is the 2N diploid state, which would be small a, small a. So if, say, the father donated the capital A, the mother donated the small a, then of course what we get out of that is a capital A, small a diploid organism. And then of course if the father donated the capital A, the mother donated the capital A, what we get out of that is capital A, diploid genotype again. Okay, so go to number 3. Number 3 says let's attach some meanings to genotypes. It says, as you see, genotype is the expression of the genetic makeup. The phenotype, as we've said before, is the actual observable result. It's really what you see from the genotype. So if you had capital A, that tree would produce big apples. If you had small a, that tree would produce small apples. So you have the genotype, which is the genetic constitution, the genes that are found, and then the phenotype would be what you actually look at is what you have there. Okay. Then if we go to the next page, they talk about human earlobes. Now, earlobes are either attached. Your earlobes, I don't know if you've ever noticed, they may attach to the side of your face, or they may just kind of dangle a little bit. That's called free earlobes, if they dangle a bit. I have free earlobes, which is actually pretty common. The attached earlobes is not as common. So it's determined by a single gene with 2 alleles, capital F, and it is possible you could have capital F, or you could have small f, or you could get a capital F and a small f, and that'd be the diploid genotypes. So an individual who has capital F, or a capital F, small f, they would have free lobes. So the dominant condition, of course, is what you have in each case. So if you have a small f, that's the only way that a person could actually have the recessive phenotype, which would be the attached earlobe, small f. So the father donated the small f, the mother donated the small f, so that would be attached lobes. So they give us a problem here. It says, let's suppose that a man has the genotype capital F. All right, so this is the man's genotype, capital F. Obviously, one of those came from his father, one of those came from the mother. What is the genotype of his gametes? Well, you know, if you wanted to run that through meiosis, like we just showed you how to do, you could. And at the end of that, you would get 4 possible gametes, but all 4 would be identical. I mean, the only possible gamete you could get out of that would be the capital F. So, you know, that's the diploid, 2N. Here's your haploid unit, which would be sperm or egg, and it just gets one of the 2 capital Fs. Okay, so the genotype of his gamete would be capital F. Okay, and it says, suppose a woman has attached earlobes. Well, she has attached earlobes. The only way a female can have, or anyone can have attached earlobes would be small f, small f. Well, if she produces an egg, the egg is either going to get this small f or the egg is going to get that small f. It's a 50-50. So the only genotype the egg would carry would be a small f. I mean, you know, that's all you have. It's going to get one of those 2 small f's. So again, this is the diploid genotype right here. And then the gametes being haploid, you only get one of the 2, so that's a haploid state there. Okay, so when both recessives are present, and we've said this before, individual is going to be homozygous recessive. Okay, so then they ask us, they say, well, suppose these 2 individuals produce a child. Well, we're saying this is the male, so that's the sperm. It's carrying a capital F in it. Let's say this is a female, which is what they said. So this is the egg. It has the capital F in it. So if we fertilize these 2, we get a union of the sperm and the egg. Then their child would get a capital F from the father and a capital F from the mother. So we restored the diploid genotype. So the child is going to have the free lobes. He will not have attached lobes. It's capital F, capital F. So the question is, the next question is, her gametes carry what allele? Would that be capital F? And then, of course, suppose these 2 individuals produce a child. Show the genotype. Sperm genotype is a capital F. Egg genotype is a capital F. The child genotype would be capital F, capital F. And, of course, that's homozygous dominant for the child in that case. It says, what is the phenotype of the child on the next page? Well, the phenotype, you know, what does it look like? Well, it's got free lobes. That's what it looks like. Okay, so then we go to number 4. It says, in garden peas, purple flowers are dominant over white. So capital P would be the allele for purple, small p the allele for white. So letter A, what is the phenotype, the color? What do these flowers look like? Well, if they have the genotype small p, that's going to be a white flower. They didn't get a capital P. They can't be purple. So that's a white flower. Capital P, well, that's going to be purple. The only way you can be white is white flower is to be small p. Purple, you got to have at least one capital P because it's dominant. So if you get both capital P's, obviously you're going to be a purple flower plant. And then the last one there is a capital P and a small p. Well, purple's dominant. The capital P is dominant over the small p for white. So even though they get a gene for purple and a gene for white, that doesn't mean they're purple and white. That just means that they're purple. The capital P, purple, dominates the white gene. It completely covers its expression completely. So you just get a purple flowered plant. Okay, then when you actually have a problem here, a genetic problem, a white flowered garden pea, well, the only way you're going to get a white flowered garden pea would be small p. Okay, small p is crossed with homozygous dominant purple, capital P, capital P. Well, this is actually pretty easy. So the only gamete you can get out of a white flowered garden pea would be small p. The only gamete you can get out of the purple flowered plant, the diploid, is this haploid, which would be the capital P. So if we join those 2 together, then of course always write the dominant gene first and the recessive second. So this individual is going to be heterozygous, is what we get out of it. Okay, so what's the questions are, what is the genotype of gametes from the white flowered plant? Well, that's a small p. That's the white flowered plant. Small p obviously is white. Okay, then letter C, what is the genotype of the gametes from the purple flower plant? Well, they're all going to be capital P. That's the only thing they can be. The parent only has capital P and capital P, so any gamete they produce is going to get one of those, either one, doesn't matter, but it's going to be a capital P. So any fertilization of the two, you would end up with a offspring with a capital P and a small p, which I believe is their last question. Oh no, letter D. What phenotype of the plant produced? Well, the genotype is capital P, small p. The phenotype, well, that's going to be purple. And again, you do get one gene for purple and you do get one gene for white, but the purple is dominant and completely just masks, covers up the expression of the white flowered garden pea. All right. Now, there is a method that you can use called a Punnett square. And what you do is just kind of write a window box on that. And you put the genotypes in the way that they say that, the genotypes of the white flowered plant, and they want that on the top. And for the purple flowered plant, they want that on the side. So then you pull these down and across. So this would be a capital P, small p, capital P, small p, capital P, small p, capital P, small p. So again, any plant that they produce would be a purple flowered plant. That's what you call the F1, first generation of plants. Actually, F stands for filial, F, well, that didn't look right, F-I-A-L. Filial means brother, sister. So any of these in these boxes, all of these in these boxes are really brothers and sisters. You'd have several P's actually produced, but if you planted all of those, coming up with purple flowers. Okay, they give us another problem. Heterozygous plant. Now, if it's heterozygous, it's going to be a capital P and a small p. Is crossed with a white flowered pea plant, small p, small p. Okay, now to run the Punnett square on that, there's two possibilities that you can get on the purple flowered plant for gametes. The gamete could either get the capital P or it could get the small p. It isn't going to get both. It's haploid, so it only gets one of those. So, and of course the white flowered plant is only going to get, it's going to either get this small p or this small p. So they want us to put the gametes for the white flower here, the gametes for the heterozygous, the one with the dominant and the recessive gene, the purple and the white on the side. So it's either the gamete would either get the capital P or it would get the small p and then pull down and across. Once you do that again, you're actually writing the dominant gene first, recessive gene second. These are both recessive, so we don't have any problem with that. So that would be the possibilities for their offspring. The genotypic ratio, I'll just write that as a GR. I have 2 capital P small p's and I have 2 small p small p's. Phenotypic ratio means what they look like. Well, these two are purple. So I have 2 purple out of that. These two will be white, and that's a ratio. So 2 purple, 2 white. Okay, well, we'll end it at that point, and that'll mean that we can pick up with number 4 when we do the lab. And that moved us forward quite a bit, which is actually a good thing, and that helps us significantly. Okay.